Hi, my name is Arizbeth Lopez. I am a junior majoring in biology with a pre-med concentration, and my mentor is Dr. Vasefi. The goal of our research project was to investigate the neuroprotective properties of CBD on Alzheimer's disease and how those neuroprotective properties will be affected in a time-dependent response. Alzheimer's disease is a neurodegenerative disease that is characterized by the formation of beta amyloid plaques. And what beta amyloid plaques are is just clumps of proteins that form between neurons and cause damage to the cells by increasing oxidative stress and ultimately leading to cell death. CBD is a non psychotropic phytocannabinoid that has gained interest due to its neuroprotective properties against A beta. Uh, the hypothesis for our research project was that CBD can increase cell viability and decrease oxidative stress. However, the effect will decrease in a time-dependent response. For our experiment, we used SHSY5Y cell line, which is a cell line that is used for different types of neurological studies, including those related to neurodegenerative diseases. We grew our cells on T25 flasks and kept them at 37 degrees Celsius, which is normal body temperature, and 5% CO2, which is used to maintain the pH of the cell culture. Once the cells were at the right confluency, we transferred them onto 96-well plates, and we plated 100,000 cells per well. After we moved the cells into the 96-well plates, we incubated them for 24 hours to allow the cells to stick to the bottom of the plate. And after those 24 hours of incubation, we serum starved the cells. And what that means is we remove the media that was already in the wells and we replace that with FBS-free media. The cells were then incubated again for 24 hours. In order to measure A beta toxicity and the neuroprotective properties of CBD, we performed MTTSA. The first picture right here under MTTSA shows the different conditions for the assay. And the way that MTTSA works, it is a colorimetric assay that is done to quantify the number of viable cells. MTT is a pale yellow tetrasol substrate that is cleaved or reduced by an enzyme in viable cells to yield a purple formazan product. The purple formazan product is insoluble, so it forms crystals, and a solubilization solution must be added to dissolve the crystals and form the purple colored solution that you see in the picture right below the conditions. The absorbance of this colored solution can be taken and measured using the H1 Synergy plate reader, and that's what we use to get our results. As stated before, A beta increases oxidative stress, and oxidative stress is related or associated with an increase in the production of reactive oxygen species. So in order to measure those, we used ROSSA. Again, the first picture shows the different conditions for the assay. And the ROS detection dye that was used is cell permeable and interacts directly with the ROS, with the reactive oxygen species. The interaction yields a green fluorescent product, which is read by the H1 Synergy plate reader. Figure 3 shows our results that we obtained from the MTTSA. This figure shows that cells treated with just CBD had an increased cell viability, while cells treated with just A beta showed decreased cell viability. Cells that were treated with both CBD and A beta still showed increased cell viability. So these results actually support our original hypothesis, which states that CBD will increase cell viability. Now for figure 4 shows the results of the ROSSA at 0 minutes, so immediately after the treatment. The The graph indicates that both CBD and A beta actually increase reactive oxygen species production in the cells. And this was not what we originally expected, so we actually looked at the results over time. And that's what figure 5 shows. The results from the ROSSA from 0 to 60 minutes. The graph indicates that ROS production of this within the cells decreased over time in the cells that were treated with just CBD. Because of this, the future direction of our project will be to study the effect of CBD on ROS production over 24 hours. We expect that ROS production will continue to decrease over time. Thank you.